Hello, everyone. Wow, it's so great to be here. Welcome to UCL's Pint of Science 2021. Pint of Science is the world's largest science festival, and every year we bring you exciting talks from around the world. Now, normally, of course, this would be happening in a pub with a pint of our favourite beer. But this year, we're coming to you live from YouTube, from the comfort of your own homes and pyjamas. I've taken real advantage of the situation. Now, we don't think that means tonight has to be any less interactive. So we want to hear your comments all the way through the show. We want them in the YouTube comments below because we've got a fantastic team slaving away and they're going to answer some of the comments. Okay. And my name's Josh Harvey. I'm a PhD student here at UCL and I'm co-hosted by the amazing E.
Hi everyone, my name is E and welcome to UCL Pint of Science 2021. And I'll be your co-host for tonight. Thank you for introducing me, Josh.
Great. We've got an absolutely fantastic lineup for tonight. It's all going to start with a talk from our guest speaker, and he's going to be telling you about sleep across the animal kingdom. And we're also going to have a chance to ask our guest speaker some exciting questions. So again, as the talk goes on, we want those questions in the YouTube comments below, and our great backstage team are going to be selecting some of our favorite questions. Now, after the questions, we're going to have a short intermission and we're going to have a quiz. And the reason why we don't want you to take the talk to heart, we don't want you to fall asleep. So we're going to give you a rapid fire 7-question quiz to fire up those neurons again. And the reason why we're doing that is we've got an exciting drawing challenge. We want you to draw your favorite animal in their pajamas. Now, of course, this can be real or it can be imagined, but we don't want you being shy because me and E will be having a go ourselves. And honestly, I am not much for a drawer and E's promised me she's not as well, so she's not going to embarrass me. We want you to upload those pictures to Twitter with the hashtag Pint21UCL. That's hashtag Pint21UCL. And we'll be picking out some of our favorite pictures and we'll be showing them at the end of the show. Okay, so I'm going to hand back over to E.
Welcome. Today we have our speaker, Professor Jason Rihel. After spending some time at Harvard, Jason joined UCL in February 2012. Jason uses zebrafish to study sleep regulation. To be more specific, it's the neuronal and genetic mechanisms that control sleep in zebrafish. Jason is not only a scientist, but also a chess player. He once participated in the underwater chess championship at the Mind Sports Olympiad. There's Jason. Hi, Jason.
Hi, Jason.
How impressive is that? But today he's going to talk to us about sleep across the animal kingdom. So let's welcome Professor Jason Rihel.
Thanks so much. I think the real trick is to not fall asleep while you're participating in the underwater chess event. That would be mighty dangerous for me, but probably not dangerous for the animal I'm going to tell you about, the model organism that we study, the zebrafish. So before I tell you about the fish and what we use it for, I'm going to tell you a little bit about why are we interested in sleep as researchers at all. I think it's something that we all have experience with. In fact, I know we all have experience with sleep, but it's a strange thing that we do when we go to sleep. First of all, it's a very vulnerable state. It's a state where suddenly we can be exposed to danger, and yet we spend so much of our time, a third of our life, this cat, maybe 20 hours a day will sleep. And we spend all this time doing this very vulnerable thing. And so it must be doing something essentially important. And yet none of the sleep researchers can agree on what that essential thing or things are. The other thing that's unusual about sleep is that unlike other things that you do in your day-to-day existence, such as even extreme, such as, well, fun things, but even extreme things like eating or drinking, you can actually stop eating to the point where you die if you wanted to go on some kind of hunger strike. but sleep is something that is unavoidable. You can't avoid going to sleep if you try to stay awake, even if you're a sleep researcher reading something fun like a book about sleep, or if you're a very motivated high school student like Randy Gardner was when he was trying to break the world record, which they don't even give anymore, for staying awake. And he tried to stay awake for many days in a row. And the point of this picture that appeared in Life magazine at the time is that his friends had to keep him occupied, like basically around the clock, in order to keep him awake. And if you look at him closely there, he looks like he's already trying to doze off. So what is it about sleep that is, puts us in this vulnerable place? And it's something that we cannot avoid to do, even if we try. And that's essentially what we're interested in to discover. I picture here the Sandman. This is the character from the Hans Christian Andersen story. And what the Sandman would do is sneak into children's bedrooms at night, and he would blow dust into their eyes that would make them sleepy, and he would tickle the backs of their necks to make them tired. And that's essentially what we're interested in as sleep researchers. We're interested in what are the molecules in the brain that are regulating sleep? What is the nature of that dust that the Sandman is sprinkling? We're interested in the neurons that are involved in controlling sleep. Where in the brain is this dust being sensed? What are the neurons that Sandman is tickling in order to drive us to go to sleep? And then on top of that, we're interested in the functions of sleep. Why do we do this? And why do we need to do this? It's completely unclear, really. So how do we study sleep? Well, in the first thing to know is that sleep is accompanied by changes in your state of your brain. And if you measure this in humans, the way this is traditionally done is you put scalp electrodes on the surface of the scalp and you record brain activity to see how the neurons of the brain are talking to one another. And if you do this during wakefulness, when a person's awake, you see this very chattery kind of pattern. This is a measure called the EEG, and you get this very chattery pattern. It would be like dropping a microphone into a cocktail party and you have a chattery kind of multiple conversations going on at once. That's essentially what your brain is doing when you're awake. You have multiple conversations going on with these neurons communicating with one another in an asynchronous pattern. But then as you go from awake into a lighter sleep stage, you start to get synchrony across the brain, where now the neurons aren't talking in this chattery way anymore. They start to talk in a kind of uniform way, to the point where you're in deep sleep and they're going through these periods of on and off states where they're basically firing in unison with one another. This might be akin to dropping a microphone into the middle of a stadium when the stadium's doing the wave and everybody is suddenly cheering in unison around the stadium in a circle. Maybe a different time scale, but it's the same idea that now all of a sudden the neurons are talking to each other in unison. And this deep sleep is often considered to be the important restorative stage of sleep. Now, there's one other state that is curiously appearing when you do this kind of recording, and that is you have these periods where the brain suddenly wakes up again, even though the person is visibly still sleeping. And this is called REM sleep, for rapid eye movement sleep, because it's one of the only places where the eyes still move. And this is when you dream, or mostly dream. For example, if you wake a person up that's in this state of REM sleep, they will explain a lot about their dream and they'll have a vivid recall of dreams. Now, using this kind of like brain activity patterns, people have thought for a long time that you can apply this to other animals, and people have applied that to birds and mammals and reptiles and so on. And people thought that they could detect very clear sleep signatures in amphibians, reptiles, birds, and mammals. But then it got a little bit dicier as you go down the evolutionary tree here. And so brain states in these animals were thought to have sleep-like states. And using this kind of brain activity has been has led to many great and interesting discoveries. For example, the idea that Some animals can sleep one side of their brain at a time. So for example, if you look at whales and you record from the left hemisphere of the brain or the right hemisphere of the brain, one hemisphere can be awake while the other one is asleep, probably allowing the dolphin or whale to go back to the surface in order to breathe, for example. They need to be constantly moving and then do the recording. So this was the state of the art for a very long time. And then about 20 years ago, some sleep researchers said, well, hold on a second. Sleep isn't just a brain state. Sleep is also a behavior. After all, human beings, long before you could record from the brain, knew what sleep was. You can even see that in the sleeping lady. This is the oldest depiction of sleep that I've been able to discover so far. This is from a museum, a picture we took in Malta from 3000 BC. And you can tell just by looking at the posture of this woman depicted here that she is very likely sleeping. And by using these kinds of behavioral criteria, such as posture, we can now see that sleep is a nearly universal phenomenon in animals. So what are some of the behavioral features of sleep? Well, it's going to be an unavoidable period of inactivity. It usually occurs at the same time every day in a species-specific way. Different animals will have places that they prefer to enter this rest-like and sleep state, such as humans going to a bed, or some animals going into trees. And then it also is accompanied by changes in your ability to respond to the environment. So for example, if I came and poked you right now when you were awake, you might say, hey, what's the big idea? Why are you poking at me? But if I came and poked you while you were asleep, I might be able to get away with it. You might not even notice. And then in addition to that, you can, if you deprive an animal or a person of these sleep states, they'll show a rebound leading to increases in sleep inappropriately the next day, for example, because they're so tired from being awake inappropriately. So let's just see a couple of quick examples of these different kinds of things across the sleep state. So species-specific posture and location. Some animals prefer to sleep up in trees with different postures. Some animals will curl up into balls. Some humans will sleep relaxed on the beach. Horses will sleep standing up. Birds can sleep, if you're a swan, on the water with your head tucked back and so on. So you got this kind of species-specific posture. You wouldn't say, this is a very distinct kind of posture as opposed to being quietly awake, where the swan might be paying attention to the environment but not really moving around very much. Vigilant, but standing still. This is a very different state. Sleep occurs with a circadian rhythm, that is a 24-hour period where you consolidate sleep to be around the same time every day. In humans, that's usually at night. And you can actually see this in this great depiction from a very famous sleep researcher. He recorded himself, his own sleep pattern, for 30 years by wearing a wristwatch that measures his activity. And you can just see some examples here of month-long traces, and you can see that his sleep is predominantly happening here the same time every day, day in, day out, and that's stable from the year 2000 to the year 2008, more or less. So it occurs with this natural 24-hour rhythm. Of course, some animals are nocturnal and will sleep predominantly at night, but humans do it sleep predominantly during the day and will be awake at night, and humans, of course, sleep at night. In addition to this, if you now deprive an animal, if you test whether they have a change in arousal threshold, you can see that, for example, there's a website called stuffonmycat.com, and you can see that this cat just seems to be oblivious to the fact that somebody has put a lot of stuff on him. And I can tell that this cat is probably sleeping because at least my own cat, if you bring out the little fingernail clippers, the cat is running for the hills and won't tolerate this at all. So they're less sensitive to their environment when they're asleep. And then the last criterion is that they have this rebound sleep after sleep deprivation. So for example, one of the classic papers in fruit flies, in fact, this was one of the most groundbreaking papers that really showed that maybe all animals have a sleep-like state. they took fruit flies. You can put them on kind of a modified tilt-a-whirl, like a little sleep deprivation chamber, essentially, that keeps them awake. And then you can see here the following day that the sleep-deprived flies are inactive and sleeping a lot more the following day than their undeprived controls. So using these criteria, we now actually believe, as sleep researchers, that sleep is nearly universal in animals. And there's been some really groundbreaking papers pushing this all the way back to some of the simplest animals like jellyfish, where jellyfish don't even have a brain. They have a decentralized nervous system, basically a neural net, and yet people have demonstrated that they also have a sleep-like state following all of these criteria. And then the related hydra, an even simpler version, also has sleep-like states. And one of the ones that I use is the zebrafish. And we and others have shown that the zebrafish also have sleep-like states following these criteria. So I focus my research on the zebrafish, and now I'm going to turn for a few minutes just to tell you why. Why would I want to study something in the fish? Well, here's the fish. This is an adult fish, and this fish can lay hundreds of eggs at once, which gives us many experimental animals to work with. And the animals develop very rapidly. So they sit on top of, here's a single cell. They sit on top of this huge yolk. This yolk fuels really rapid development so that the cell divides many times, very quickly, basically every 15 minutes in the first stages of life, such that by 24 hours, you already have a pattern animal. You can already see the brain has started to develop. You can even see the eye is starting to form here. This is, this was a single cell just 24 hours before that. And then 5 days later, the fish is a fully patterned animal. It's capable of doing complex things, such as sleeping. Here's what it looks like. This was a fish. This fish was a single cell just a few days ago, and now it's able to swim around and do complex things like hunt that paramecium. You can see it going down its gullet. The other great thing about the zebrafish is that they're optically transparent. So you can put, for example, a green fluorescent protein into their brain, and then you can just image straight down into their brain, and you can see basically the brain sitting here inside the nervous system of this fish. So this is a really nice way to be able to look at brain imaging. And just to highlight that in a little bit more kind of power, we're going to now dive down through the fish brain with a 2-photon microscope, a special microscope that allows us to have very high resolution. And we can dive deep down into the brain. You can see basically as we dive stacks up and down the brain and we change the focus, we can see deep into the brain. The nuclei of the neurons are kind of these black circles, and the fluorescent part of the neuron is the cell body of the neuron, which we can see basically every single cell of the brain. Here, for example, we're zooming in on a part of the brain that's responsible for vision. The other great thing about the fish is that we can track them very simply using a video camera. We can put them in an 96-well plate. They're small, and so you can do this kind of high throughput tracking. And in systems in my lab, we track about 1,000 fish of this age at a time. And we can use the video cameras and automated software to track when the fish are active and inactive. I'm just highlighting here what it looks like. You can see they have a lot of room to swim around in, and every time they're moving, the camera or the computer is automatically detecting this with a little red flash. And if you plot what a single fish looks like over a day-night cycle, we can shine lights on them and show them different days of patterns. You can see that they're active during the day, like us, and then they're less active at night. And this is a single fish here, so you can see that there's some variability in the pattern, but they're more active during the day and less active at night, like this. Okay, so in the last part of the talk, I want to say, well, what are some of the kind of research questions that we're addressing in the zebrafish? And one of the things that we're interested in is trying to identify neurons that are responsible for driving sleep and wake states. And I'm going to tell you a little bit about one of those critical sleep-wake neurons that we know is important in humans and also seems to play a role on regulating sleep in zebrafish. And that, to introduce that, I want to introduce you to a dog. So here's a dog, Rusty, I believe his name is, and he's very happy, and he's out about, he loves to play, but he gets excited and he immediately falls into a sleep-like state. Turns out that Rusty has narcolepsy, which is a sleep disease that humans also get, in which your sleep-wake states are unstable, and you can fall dramatically like this, especially when you're excited about something like Rusty was playing out in the wild. Researchers often use palatable food. If you give the dogs like that wet food that comes out of a can, the dogs will get very excited and they'll lose posture control and they'll enter some sleep-like state like this, a cataplectic attack it's called. But you also have unstable sleep states. And so narcoleptics also will have transitions at night more rapidly. So they'll fall asleep, but then they'll wake up and fall asleep and wake up, and it's an unstable sleep-wake state. And just highlighting here, there's a particularly good and famous example where River Phoenix played a narcoleptic character in My Own Private Idaho movie from, well, some time ago now. So we now know a lot about how narcolepsy is caused in humans and dogs. And its cause is the loss of a special set of neurons in the brain. These neurons sit in the hypothalamus, so deep in your brain. This is an image of a brain slice through the mouse, and we've labeled here these neurons. They're called hypocretin neurons. It also has the name orexin. So these orexin hypocretin neurons sit here in the hypothalamus. This is a neuropeptide that these neurons secrete in order to drive wakeful states. And what happens in narcoleptic patients is that somehow these neurons die and disappear. And now all of a sudden you don't have these cells and you have narcolepsy. So some years ago, we showed that the zebrafish also have these hypocretin neurons. So here I am looking down into the zebrafish brain. Instead of having 1,000 of these, like the mouse brain does, the hypocretin neurons, there's about 10 of them in the zebrafish, but they're in the same place. They're in the zebrafish hypothalamus. So the fish, even though they're diverged from humans millions of years ago, they still have these critical neurons. If you use different genetic tricks to put that green fluorescent protein into them, you can actually trace where these neurons connect in the brain. And we see that the zebrafish neurons connect to many of the same places that we know are connected in humans. So for example, if we follow these tracks here down into this one area that's labeled in red, this is an area called the locus ceruleus. This area produces norepinephrine and is a major center of driving arousal in humans and in rodents and also in zebrafish. And you can see that these send these nice projections there and connect to them. In addition to that, because the fish is optically transparent, we can watch neuronal activity while the fish are behaving. And so you can see, for example, if I now put in a special sensor here, this is going to sense calcium that's released when neurons are active. And we're going to watch the fish using a microscope, and we're going to record their tail activity with a camera so we can see when the fish is moving and not moving. And then we're going to look at the activity of these hypocretin neurons. And because these neurons are responsible for driving and maintaining wakeful states, what we see is that when the fish is awake by the tail, you can see the tail's moving here in this red. You can see that these neurons are sending all these kinds of, they're very active and they're showing these different calcium signals. Each of these traces is a different neuron. But you can see at night, these neurons become quiescent, they stop firing, and you can see the fish is also sleeping during this state where there's a little bout where they kind of toss and turn a little bit, but they're mostly sleeping in this state. So these neurons are active during wakefulness and they're responsible for maintaining the waking state. Now, finally, we can manipulate the hypocretin levels in the brain and ask, how does that change waking behavior? And we can use this trick where we use a special promoter where we drive hyperactivation of hypocretin using what's called a heat shock promoter. Basically, you warm the fish up and it will now produce a lot more of this peptide. You can see here, if I label the hypocretin, here's a normal fish. You can see the hypocretin neurons there and the projections in red. And you can see after we flood the brain with hypocretin, a massive flood of hypocretin using this trick comes about. And then we can track how their behavior changes when you do this. And if we do that, you can see that they still have this normal sleep-wake pattern where they're active during the day and less active at night, but they're way more active. You can see several times more active at night. This is in the red after we flood the brain with this peptide, and it lasts for several days where they're just more active. And so using this and other kinds of research and other kinds of tricks, we and others have come up with basically this idea that you have these kind of wake sensing or wake promoting areas in the brain and sleep promoting areas in the brain. They mutually inhibit one another such that when one is active, the other is inactive. And that the hypocretin neurons, these orexin neurons, are somehow promoting wakefulness by kind of pushing the gas into the wake state at the expense of the sleep state. And by doing tricks like this, we're trying to unravel basically more about this mechanism, how the brain actually senses sleep need and turns it into sleep behavior. So just to summarize, I've told you that sleep is a universal feature of animals when we measure it as a behavior. I think the zebrafish are a great neuroscience tool for a variety of the different reasons that I proposed. And zebrafish and humans share at least one critical sleep-wake population of neurons, and we've been discovering many others that we think are important. With that, I thank you for your time and attending, and I'm happy to answer questions. The end.
Thank
Fantastic.
you, Jason. That was wonderful talk. I've learned so much, and it's just truly fascinating. The fluorescent pictures look amazing, though, by the way.
Thanks.
And the cute cats. We've got comments about the cute cats.
The internet is made of cats, so you can always get nice cat pictures that are sleeping.
Yeah, indeed. So we're going to have a short intermission soon before we proceed to the Q&A session. So I will say goodbye for now, Jason. See you in a minute.
See you in a minute.
Bye, Jason. He's off for a quick power nap, I think.
Yeah, I saw that.
So now's your chance to get your questions in for Jason. We've had some fantastic questions so far. I've had a look at some of them, and I think he's going to be hard pushed to answer some of them because they seem really challenging. But I do want to remind you about the drawing challenge before we go to our intermission. We want you to draw your favorite animal in their pajamas. And of course, it doesn't have to be real. You can take inspiration from anywhere, and don't be shy because we'll be having a go ourselves. And the way you submit them is upload them to Twitter with the hashtag Pint21UCL. That's hashtag Pint21UCL. We've got a short intermission now, so plenty of time to dust off those drawing pencils and submit your picture. Okay, so that's everything for now, and we'll see you soon. Okay, bye.
Bye. Hi, welcome back, everyone. And welcome back, Jason. Where's Jason? Hi.
Here he is.
Hello. I'm awake.
Waking up from his nap.
Yeah. So now we're on to our Q&A session. Because of time constraint, we'll try our best to get through all your questions. So Josh, do you want to start with the first one?
Yeah. Okay, Jason, I hope you're ready for these. So the first one we've got from Jellyhead143. And the question is, how come having too much sleep makes you more tired?
Yeah, I am really interested in this question. And I don't think we know the answer to this, nor do we have a lot of research studying this. I suspect that's because it's a lot easier to force somebody to stay awake than it is to force somebody to sleep longer, right? You would have to use some kind of drug or some kind of other intervention, but then drugs don't produce a natural kind of sleep, and so it would be very hard to study. So I know very little research on this, although I myself I really want to know the answer to that, but I don't know actually.
Yeah.
Well, now the second question is from Natalie. Are there any animals that don't sleep at all?
So, to the best of our knowledge, all animals that have been tested carefully seem to have a sleep-like state. Now, some animals, this seems restricted to only certain periods of their life, like during development. So, for example, the roundworm C. elegans its predominant mode of sleep happens during a very specific kind of developmental window when they shed their cuticle and molt. But so far, when we look carefully using these behavioral criteria, we find sleep-like states everywhere we look.
That's interesting. So pretty much conserved across the animal kingdom, I guess.
Seems to be. If it's in a jellyfish, it's probably going to be pretty broadly applied, I think.
True.
So our next question is from Paddy Holroyd, and it's a question close to my heart because I love these on the weekend. How effective are power naps? So a quick 20-minute nap. Are they worth doing?
So a lot of research suggests that they are very effective, and there's a variety of different ways that people have studied this. For example, impacts on learning and memory. A short nap can improve, for example, learning on different kinds of mechanical motor tasks and other kinds of tests. There are some diminishing returns. I think there are some cases where if you try to do a power nap too late in the day, then there can be some impacts where now you might get some insomnia where you're just not tired, you're just not sleepy enough in the evening when you're ready to go to bed. But almost all research seems to suggest that power naps are great in terms of cognitive performance, long-term health consequences, and things like learning. Yeah.
Great. That was my suspicion because I find them so good. So I thought they must be useful.
All right. So next question's from Phil. His question is, I have suffered with insomnia and have been told that breaking up my sleep into 2 parts over the course of 12 hours is better for you, especially you're struggling with sleep. So how true is this?
Yeah. So as a caveat straight away, I'm not offering any medical advice. So I recommend you talk to a sleep physician if you want good medical advice about insomnia. However, what I can tell you is that the general advice is very often that if you're struggling to fall asleep, if you don't fall asleep after 20, 30 minutes or so, the general advice is to stop trying to sleep, get up, be quietly restful, do something restful and calm for 20, 30 minutes, and then try again. This ties into some interesting research, historical research, in which some historians have suggested that sleep in humans used to be broken up into 2 phases, in which there would be kind of like the sun would go down, there wasn't any electricity, and so the sun would go down, and you would sleep for a while, and then you'd get up for for maybe an hour or so in the middle of the night, and then have a second sleep later after that. And so this advice about breaking it up may be related to this kind of historical idea. However, that's controversial. Some people who study, for example, indigenous tribes in South America, where electricity still isn't really used, have found conflicting results. They don't see that humans that are like living in a non-electrical environment break it up into two sleeps. They seem to have one single consolidated sleep. And so this idea, although there's some evidence from the historical record about it, it's a little bit controversial on whether this is really truly a natural human state.
Interesting. Sounds really like a complicated topic.
Absolutely. Yeah, absolutely.
So our next question is from Gil Polgreen. Do coastal birds sleep at the same time each day, or does their sleep rotate with the tide so they can feed? He's obviously thought she's obviously thought about that. That's a clever question, I think.
That is a very clever question. And I have to admit that there are a lot of really fascinating studies with coastal birds. One example is the sandpiper. The sandpiper males, they actually change their sleeping schedule with the breeding season. So for example, sandpiper males will essentially sleep deprive themselves for many, many, many days and try to mate with as many females as they can. Some evidence suggests that some of the males, the most successful ones from an evolutionary point of view, because they have the most offspring, are the ones that go without the most sleep during this period. So, but I don't know about the feeding and the tides. However, I do know of another example, which is really fascinating, and that is bats. So bats are mostly nocturnal, right? Everybody thinks about the bats coming out at night. But bats actually, or at least some species of bats, will actually shift their activity and sleep patterns depending on the availability of food. So when your days are long and your nights are short, versus when your days are short and your nights are long, as the seasons change, the insects come out at different times, and the bats will shift their sleeping in order to match when the insects are coming out. So I suspect the coastal birds do something similar, but I don't know off the top of my head if that's been shown for sure or not.
Wow, that's so cool. Okay, so our next question is from David. He's asking, why are the old insomniacs?
So this is a question about why do people so if you look at a plot of sleep as people age, sleep gets shorter and shorter, and people that are old tend to sleep a lot less. I don't think we know the full answer to this, but there's some evidence to suggest that some of these sleep promoting areas in the brain that I'm talking about start to degenerate when you're older, and so therefore you have fewer neurons. So for example, there's this sleep promoting area in the brain that expresses a different neuropeptide from the hypocretin. It's a lot about neuropeptides. They produce this neuropeptide called galanin, and there's been some studies that show that the degree of loss of galanin neurons can correlate pretty well with the amount of sleep loss. I'm leaving aside the link between sleep and Alzheimer's disease. There's some suggestion that short sleep leads to the buildup of some of the toxic byproducts that contribute to Alzheimer's disease, and then therefore there's an idea that those toxic byproducts might be damaging the very sleep-wake centers that we're talking about, leading to shorter sleep, leading to more buildup of toxic byproducts, leading to more damage, leading to more shorter sleep, and so on. You get this vicious cycle. So that's also another component of sleep and aging that is a really hot topic of research right now.
That's interesting. So we've only got time for one last question, I'm afraid. We've had so many questions in, so apologies to anyone we haven't been able to get to. But the final question is from JD111. Has human sleep evolved as we have become safer during sleep with no predator threats apart from snoring bedfellows? Great question.
So that is challenging. I think the short answer is that humans yes, it's true that we have no predator threats now, but I think if you talk to people who do human evolution, they will tell you that that's a very recent phenomenon, right? Like civilizations and cities and stuff don't even exist until 10,000 years ago. That's really not a long enough time scale for evolution to have major, profound effects on too much behavior. There's some examples like lactose tolerance and things like that, but I think probably the time scale hasn't been long enough yet. But it is true that the level of predation affects your, has evolutionary effects on sleep in other animals. So animals that are at the top of the food chain sleep a lot more, right? Like there's a reason why a lion sleeps 20 some hours a day and a giraffe sleeps only a few hours a day, for example. I mean, that is definitely true, that evolution sculpts this. There's actually some fascinating evidence that that suggests that that this also happens in some fish species as well. So fish that live in predator-rich environments versus predator-poor environments will shift their sleep patterns in order to avoid the predators.
Great answer to a great question, I think.
Yeah, so that is the end of our Q&A session. Thank you, Jason. Thank you for your wonderful talk, and thank you for answering all the questions.
Yeah,
Thanks,
I'm happy
Jason.
to answer anything. Thank you so much.
Bye.
Bye. Thanks very much.
Okay, so now we're moving on to our quiz session. So, Josh, would you like to introduce the quiz?
Yeah, I'm ready. I hope you're ready, and I hope everyone's ready at home. But before we begin, I just want to remind everyone to keep submitting your pictures for the drawing challenge. Upload them to your Twitter with the hashtag Pint21UCL. We want to see your favorite animal in their pajamas. Okay, and to get you warmed up for that drawing challenge, let's have this 7-question rapid fire quiz. Okay, so let's begin. So the first question is, when falling asleep, our bodies can suddenly jerk and it can feel like we're falling. What's this sensation called? Is it A, a hypnic jerk, B, a falling jerk, C, a haptic jerk, or D, a snoring jerk?
Snoring jerk. It sounds really funny.
And the answer is actually A, a hypnic jerk. This is an involuntary movement that's often made when someone goes from an awake state to an asleep state. You may have seen it yourself, or watching someone as they fall asleep to suddenly shake like that. So I hope you're keeping track of your scores at home.
So the second question is, what is the official term for drugs prescribed to induce sleep? A, narcotics, B, hypnotics, C, antidepressants, or D, anesthetics? Which one would it be? The answer is B, hypnotics. In fact, the term originates from Greek, so the hypno means sleep. Next question.
What is the scientific term for the study of dreams? Is it A, morphology, B, enology, C, oneirology, D, dreamology? What do you think?
Dreamology sounds really fairy tale.
Seems too easy though. So
True.
the actual answer is oneirology. This term became more widespread when people started using EEGs to measure sleep-wake cycles. And they found something you might have heard of before, REM, which is where your brain signals are very similar to the ones you have whilst you're awake.
So the next question is, which of these animals sleep the least per day? A, goat, B, albatross, C, fruit fly, or D, lizard? What do you think, Josh?
I've got a guess, but I'm about 25% sure I'm right.
Oh, the answer is here already. So it's B, albatross. In fact, they only sleep 40 minutes per day. Very little sleep.
Does human sleep synchronize with the lunar/moon cycle? Is the answer A, yes, we sleep less during a full moon, B, no, C, yes, we sleep more during a full moon, or D, maybe? Hedge your bets, you're not sure? And the answer is yes, we sleep less during a full moon, and it's thought the brighter light from a full moon means we go to sleep later and sleep for a shorter period.
Which of these animals can sleep standing up? Select all that apply. A. Horse, B. Giraffe, C. Bear, or D. Elephant. Remember to select all that apply.
Was that a hint, E?
Yes. So the answer is A, B, and D. So bear here, only bear doesn't sleep standing up here. Next question.
So this is the last question. Which of these marine mammals can sleep unihemispherically, which means one side of the brain is asleep whilst the other is awake? Is it A, killer whales, B, dolphins, C, eared seals, or D, manatees? What do you think? Last question.
A tricky one, though.
Yeah. And it was a bit of a trick question, I'm afraid, because the answer is A, B, C, and D. And Jason mentioned this during his talk, but these animals sleep with one side of their brain only. And that means if you watch them sleeping, they'll actually just have one eye open. Isn't that creepy? So that was question 7. We want to hear what you got at home. So in the comments below, can you post your scores? We want to see if anyone managed 7 out of 7.
Yes, so keep the scores coming in. And we're moving on to the last, but I think it's the most inspiring part of the night. So let's have a look at the drawing challenge. And here's what I drew earlier.
That's very good.
Yeah,
you told me you couldn't draw.
Totally lied.
That's really good. Yeah, that's amazing. Looks completely passed out there.
So what about you, Josh?
So I've gone down the primate theme, but the monkey's ready for bed, definitely.
That is really cute.
And I've just noticed he's missing a bit of his trouser there.
That's
but
okay.
Never mind.
Doesn't need a trouser.
Well, should we have a look at what people have drawn at home? I'm really excited to see what they've drawn.
Yes, let's have a look.
Oh, that's fantastic. I like the cat. The cat looks real in their little with the little hat, bit like the monkey hat, to be honest. But yeah,
you just like the hat, don't you?
Yeah, I think so.
I quite like the bird. It's like she or he looks like a professor there.
Yeah, it does.
The pajama looks like a suit.
Yeah, at Pingu University. Looks great. Should we see a couple of more?
Yes.
See a couple more. What do you think? These are good. I like the giraffe. I think that's might be my favorite so far, actually. It's just got a bit of a cheeky expression.
Really? Yeah, it's really cute, but I quite like the fish as well.
Yeah.
But it's missing pajama. I guess fish don't sleep with pajama.
Probably not.
Mm-hmm.
Well, should we have, I think we've got time for just a couple more. Should we have a look at them?
Yes.
Fantastic.
Nice drawing.
So what's your favorite so far?
I like the goldfish. I used to have goldfish as a pet when I grew up. What about you?
Very good. I think I'm sticking with the giraffe. There was something about the giraffe that I just really enjoyed.
Nice.
So that's everything we've got time for. I thank you so much for coming. I hope you've enjoyed the talk and you've enjoyed listening to Jason and having a chance to ask him some questions. We've been having some great interaction with the audience in the comments, but we'd like to hear what you thought of the show. And below in the YouTube description, you'll find a link to a feedback form. And it'll be great for us to hear what you thought of the show. And also it gives you a chance to win a Pint of Science pint glass. And that's not even something I've been given or E's been given. So it's a fantastic opportunity and we'd love you to fill that out. What do you think, E?
I will fill it out as well, I think.
Yeah, I want a chance to win that pint glass.
Yeah, it looks good. I want it. Okay, so that's the end of our show. Any more to add to that, Josh?
No, I think I'm ready for bed. Actually,
Wow.
I'm just gonna. That talk and the quiz and the drawing challenge. It's just really tired me out. So I think I'm ready for bed.
Me too. So that's it. Thanks for joining us, everyone. Bye bye. Sleep tight.
Bye, everyone. Sleep tight.